Hi guys, Miss Francis here. Today I would like to talk to you about the endomembrane system. We learned that eukaryotic cells are compartmentalized. Each one of these different compartments is a different organelle, and each organelle has a unique function. However, these organelles do work together in order to create proteins and get those proteins where they need to go. This system is known as the endomembrane system, and it includes the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, the lysosomes, the vacuoles, and the plasma membrane. So let's get into the nitty-gritty as far as what exactly is going on. So first we start with our DNA. Our DNA is found in the nucleus, and it's going to contain the blueprint or the instructions for making a protein. But DNA is too big to travel outside of the nucleus to bring the message to the ribosomes. So the DNA must send its message in messenger RNA. And that messenger RNA carries the code for protein synthesis to the ribosome. The ribosome is actually made by the nucleolus, and it's at the ribosome that the protein is going to be synthesized. Now, the rough ER is like the highway system of the cell. It's going to allow that protein that my ribosome just made to travel, and together with the smooth ER, it's going to enable that protein to get wrapped inside a vesicle. The smooth ER and the rough ER together are known as the membrane factories, where the smooth ER is going to make the phospholipid and the rough ER is going to make the protein. Once that protein is wrapped inside the vesicle, it travels to the Golgi body via a transport vesicle. At the Golgi body, that protein is going to get modified, it's going to get tagged, so it's going to get carbs added to it or lipids added to it, giving that protein its unique shape and its unique function. So the Golgi body gets it ready to be shipped. It's like the post office. It's going to modify, it's going to sort, and it's going to identify where that protein needs to go. Well, how does that protein get to where it needs to go? It's going to travel in a secretory vesicle, which is going to carry the protein to the plasma membrane. Once that secretory vesicle is at the plasma membrane, it's going to fuse with the membrane, and through the process of exocytosis, it enables that vesicle to bud off and release the protein that's inside of it to the cell. Now, what if I needed a protein for use inside the cell? Well, in that case, instead of being packaged in secretory vesicles, they are going to be packaged into lysosomes, which are special vesicles. So for example, digestive enzymes are needed within the cell. So instead of those digestive enzymes being packaged in secretory vesicles and then fusing with the plasma membrane, those digestive enzymes are going to be packaged into these lysosomes. Here are some great videos on how the endomembrane system works, and we're going to actually use this video as a supplement to our notes. So if you get a chance, head back and view that video. You're going to need to know the steps for making a protein. In other words, where is that protein used? So what's the process of making that protein? For example, insulin. Insulin is going to be used outside of the cell. So the pathway of insulin isn't going to include lysosomes. It's going to include secretory vesicles instead. So let's go through the pathway of insulin through the endomembrane system. Well, once again, we start out with our DNA. The DNA codes for making the protein. In this case, our protein is insulin. Insulin is a hormone. A hormone is a protein. That code is in the nucleus. That DNA then sends the message via messenger RNA. The DNA is transcribed into messenger RNA through a process known as transcription. That messenger RNA then leaves the nucleus through the pores that are in the nuclear envelope and gets translated into transfer RNA. Once that transfer RNA hits the ribosome, it's at the ribosome where our proteins are going to be made. Now, the ribosome itself is made by the nucleolus, but at the ribosome, that's where the proteins are going to be made. In this case, our protein is insulin. So the rough ER serves as the highway by which that protein is going to travel, and together with the membrane provided by the smooth ER and the membrane that is already there in the rough ER, it's going to bud off into this transport vesicle. So now this transport vesicle is carrying the protein, the insulin, and has a membrane surrounding it. It's going to head to the Golgi body, because at the Golgi body, that's where we're going to package, we're going to receive and modify and then ship the Golgi body out to the plasma membrane. The Golgi body are composed of cisternae, which are just interfolded network of tubes where the space within the tubes is known as a lumen. The cis end of the Golgi body is what receives the protein, and the trans end of the Golgi body is what's going to ship the protein via a secretory vesicle and head to the plasma membrane. And then at the plasma membrane, that vesicle is going to fuse with it and bud off and enable the insulin to travel within the body to get to where it needs to go. So that's our discussion of the endomembrane system. Next time I see you, we'll be discussing the nucleus and ribosomes in greater detail. See you then.